Okay, so first, let's talk about the syllabus. So those of you who have been following the syllabus might have noticed that the end of next week, there's, in the tentative class schedule, there's a test schedule. Okay, now we're a little bit behind the syllabus. Here we are, February 5th. We should have started cell structure. We might start that today. We're just a little bit behind the syllabus. So what I'd like to do for test one, I think we should move that to Monday, February 15th. So next week, we should be able to finish up cell structure and cell membranes, probably start cell membranes. So we should continue, let me see, so if we start cell structure today, we'll continue with cell membranes on Monday. We'll probably finish cell membranes and start cell structure, rather the other way around. We'll finish cell structure, start cell membranes on Wednesday. This coming Friday, we're off. Okay, it's one of those non-instructional days. That means that there'll be no labs next week, because obviously if we can't have the Thursday lab, then we can't get the labs out of sync. So there'll be no lab next week. Then Friday of next week, which is, that would be the 12th. Usually what I do before a test is I'll give you a review day. Okay, so on the 12th, we won't cover any new material. I'll have a study guide available sometime before Friday the 12th. So on the day of a review, I'd like you to look over the study guide before you come to class and come with questions ready. I'll be happy to answer all of your questions on anything we've covered for the entire period. If there's something that you're not sure about that you'd like me to go over again, we could do that as well. Okay. Another thing, quizzes. So remember, quiz number 3 will be online by about noon today. That'll be due by 11:59 p.m. this evening. Another thing that I didn't cover as far as quizzes go, one thing that I'll do is I'll take one question off every quiz and put it directly on the test. All right. Now, you might recall that I said the test is going to be 40 multiple choice questions worth 2.5 points apiece. And for each quiz, there's only one multiple choice question, then there's 2 fill in the blanks and 1 true or false. Okay. That doesn't mean I'm always going to take the multiple choice question from the quiz and put that one on the test. Okay. I might take one of the fill in the blanks, turn that into a multiple choice question, put that on the test, or I might take one of the true false questions, again, turn that into a multiple choice question and put that on the test. But it's too bad we didn't fit more quizzes in. Of course, we had technical difficulties. Too bad we didn't fit in more quizzes before the test. But if you study your quizzes, then for each quiz that we fit in before the test, that's at least one question that you should be able to get right. Okay. Also, the tests this semester are going to be in seat. So unless you can't be here due to quarantine or illness or some other reason, I will expect you to be here in class on Monday the 15th for test number 1. Okay. Any questions on that? Any questions from anybody joining by Zoom today? Okay. If there's no questions, let's continue with the PowerPoint presentation. Okay, so when we had left off on Wednesday, we were talking about lipids. So recall that this PowerPoint presentation is on the 4 molecules of life, or biological molecules. There are 4 molecules of life. We have carbohydrates, lipids, nucleic acids, and proteins. So we already talked about carbohydrates. We had just started talking about lipids, so there's a couple, there's 3 different types of lipids we're going to talk about. We finished talking about the first type of lipid on Wednesday, and those are fats. So well, let me ask you guys. So there's basically 2 parts to a fat. Can anybody tell me what those 2 parts are? So we have one thing that kind of acts like the backbone, and then we have either 1, 2, or 3 of the other thing. Anybody recall? What's that? I'm sorry. I still couldn't quite make that out. Round, did you say? That's getting more into, there is such a thing as brown adipose tissue and white adipose tissue, but that's made up of these types of subunits I'm talking about. So, let me get the drawing tool. So the 2 things I'm talking about, fats are composed of a glycerol. Okay, a glycerol kind of makes up the backbone of the molecule. So the glycerol is what we see in kind of a greenish, like teal color right here. I don't know why my computer keeps doing this. Sorry about that. That's some kind of a glitch that keeps happening. Okay, hopefully it'll behave for me this time. Okay, so in this kind of a greenish teal color right here, this is the glycerol backbone. Okay. Then a fat has either 1, 2, or 3 fatty acids attached to it. Okay, the fatty acids are, so we have this long hydrocarbon chain with this carboxyl functional group. Okay, that whole thing is a fatty acid chain. So I mentioned that there's 3 different kinds of fats. So this one in particular, we have a fat with 3, that's a glycerol with 3 fatty acids attached to it. What was the name I used for when we have 3 fatty acids attached? Triglyceride. Okay, and if we have 2 attached, diglyceride, and 1 attached, of course, that's a monoglyceride. Okay. Again, just to review, there's 2 types of fatty acids that we can have in a fat. We can have a kind where they have the maximum number of hydrogens and no double bonds. And then like the bottom here, you can have carbons with double bonds, and these types don't have the maximum number of hydrogens. So what's it called when we have the maximum number of hydrogens and no double bonds? What kind of a fatty acid is that? Saturated. Okay, saturated fatty acid has the maximum number of hydrogens and no double bonds. How about, so look at this last fatty acid down here. We have a double bond that puts a kink in the hydrocarbon backbone of that fatty acid. What do we call that? Unsaturated fatty acids. Okay, very good. And I think the very last thing we went over before the end of class on Wednesday was that we're going to have different properties. Fats are going to have different properties based on whether they contain mostly saturated or mostly unsaturated fatty acids. Okay, so remember, plant fats, things like canola oil, peanut oil, usually the types of oils that we fry things in the kitchen, they have mostly unsaturated fatty acid chains. So because unsaturated fatty acid chains are kind of bent and kinked, that prevents these fats from packing together efficiently. And that's the reason why these types of fats tend to be liquid at room temperature. So if you look in your kitchen, any type of a plant fat, like canola oil, vegetable oil, peanut oil, what other kinds are there, sesame oil, all of these things are liquid at room temperature. Okay. Animal fats, on the other hand, have mostly saturated fatty acid chains, which are straight. Because these fats have straight, linear fatty acid chains, they can pack together very efficiently. This makes animal fats solid at room temperature. So again, if you look in your kitchen, any fat that comes from an animal source, like butter, like lard, these are going to tend to be solid at room temperature. Okay, so I think that about does it for fats. If there aren't any questions, Let's continue on. Okay, so the next type of lipid we're going to talk about are phospholipids. Phospholipids are similar, very similar, I think, to fats in their composition and their structure. Just like with the fats, we have a backbone made out of glycerol. So in this drawing, the glycerol backbone is right here, okay? Just like with fats, we have fatty acids. So in the case of a phospholipid, we have two fatty acids. So here's one, we have one saturated fatty acid chain, we have one unsaturated fatty acid chain. The only difference, instead of attaching a third fatty acid, we have a hydrophilic phosphate group, which is right here. Okay? That's the only difference. Now, phospholipids are the main components of cell membranes. So a cell membrane is the outside of a cell. It's the barrier between the extracellular environment and the intracellular environment. Okay? Now, let's talk about a word on this slide that you might not be familiar with. So we have one glycerol backbone, we have two fatty acid chains, and we have one hydrophilic phosphate group. Does anybody know, has anybody seen that term before, hydrophilic? Yes.
Yeah, it's attracted to liquid.
Attracted to liquid, you said? That's a pretty good definition. The liquid we're talking about specifically is water. Okay, so hydrophilic, if we look at the Greek root of this, basically means water loving. So anything that's hydrophilic tends to be soluble in water and attracted to water. So it wants to be in it wants to be around water. It wants to be in an aqueous environment. Okay. Now the other side of this coin is hydrophobic. I'll tell you what. Let's go to the whiteboard. And let me get my own dry erase markers so we're not cross-contaminating and spreading germs. Okay, so hydrophilic and hydrophobic. So first, hydrophilic. Okay, literally, this means water loving. Hydrophilic molecules are soluble in water. Chemically, hydrophilic molecules tend to be polar or charged or both. Okay, so you should be familiar with these two terms from chapter 2. Charged would be carrying a positive or negative charge, like the ions that we talked about. Polar, of course, means that it contains polar covalent bonds. So with polar covalent bonds, we have partial negative and partial positive charges that can form hydrogen bonds. That also makes molecules hydrophilic. Okay. Now let's talk about the other side of the coin. The other side of the coin would be hydrophobic. Hydrophobic molecules, so literally the word hydrophobic, if we look look at the Greek root of the word, means water fearing. Molecules that are hydrophobic are insoluble in water. Okay, and chemically, if we look at the properties of hydrophobic molecules, they tend to be uncharged and non-polar. Okay, so molecules that are hydrophobic don't want to be in an aqueous environment. They want to be away from water. Molecules that are hydrophilic or water-loving, they want to be in an aqueous environment. They want to be around water. So a good example of this is, again, we're going back to the kitchen for an example, is Italian salad dressing. If you look at Italian salad dressing, before you mix it up, you can see there's the vinegar at the bottom. Okay, vinegar is just a solution of acetic acid. Okay, vinegar is about 5% acetic acid, so it's mostly water. 5% acetic acid means it's 95% water. Vinegar is mostly water. Okay, the olive oil in your Italian dressing, well, that's a fat. Okay, that's made out of triglycerides. Okay, fats are hydrophobic. They don't want to be around water. Vinegar, of course, is mostly water. So before you shake it up, you've got the fat floating on the top and the vinegar floating on the bottom, and sinking to the bottom, rather. So that's an example of a hydrophobic versus a hydrophilic molecule. Okay, let's go back to the PowerPoint presentation. All right, there's one more term, and I'll put this on the whiteboard too, that I want to talk about before we continue. So, again, phospholipids are composed of one glycerol, which is here, two fatty acids, here and here, and one hydrophilic phosphate group. Okay, if we look at this phosphate group, we can see a negative charge here. This oxygen double bound to a phosphate, that's a polar molecule, positive charge here. So this is hydrophilic. Let's look at the fatty acid chains. Okay, so looking at these fatty acids, carbon and hydrogen have pretty much the same electronegativity. So these are non-polar covalent bonds. These are regular covalent bonds. Okay? And we don't see any positive or negative charges. Do you think this would be hydrophilic or hydrophobic? Hydrophobic, okay? So the fatty acid chains here and here are hydrophobic. And the phosphate head, sorry about that. Okay, the phosphate head over here is hydrophilic. So we have a special word for molecules that have both a hydrophilic component and a hydrophobic component. Okay, and that word, let's go back to the PowerPoint, the whiteboard rather. That word is amphipathic. So amphipathic means having both hydrophobic and hydrophilic components. Okay, amphipathic, having both hydrophilic and hydrophobic components. So if we have something like a phospholipid with a hydrophilic phosphate head, two hydrophobic fatty acid tails, that makes a phospholipid an amphipathic molecule. Okay, let's go back to the PowerPoint. All right, and the last point on this slide, these are the main components of cell membranes. When we start chapter 4, in chapter 4, we're going to talk all about cell structure and cell membranes. So we'll talk exactly about how phospholipids can form a cell membrane next chapter. Actually, we're going to talk about it now. Sorry, I'm a liar. I didn't know that, I forgot that this was in here. Okay. So phospholipids can form phospholipid bilayers. Okay, and cell membranes are made from phospholipid bilayers. Okay, when we put phospholipids in water, they will spontaneously form into these phospholipid bilayers. So let's look at this structure. So this is a phospholipid bilayer. You can see the individual phospholipids in here. So the yellow part is the phosphate head. The little white tails, these are the fatty acids attached to the glycerol. Okay? And you can see they form this phospholipid bilayer. So the fatty acid tails, they're hydrophobic, right? So are they going to want to be around water? No. If you look at the phospholipid bilayer, you can see they're sandwiched in between away from the water. Okay, so by making this phospholipid bilayer, they're in the middle of the sandwich away from water. Okay. Now the phosphate heads are hydrophilic, right? So they're going to want to be around water. So the phosphate heads are on the outside here, interacting with water, and they're on the inside here, interacting with the water in the middle of this phospholipid sphere. Okay, so this is a phospholipid bilayer, and cell membranes are composed primarily of phospholipid bilayers. Okay, here's a little bit of a bigger blown up drawing of a phospholipid bilayer. Okay, so again, we have water here, water here. Again, we put phospholipids in water. They will spontaneously form into this phospholipid bilayer. The fatty acid tails sandwiched in between, away from the water in a hydrophobic environment. The hydrophilic heads on the outside are interacting with water. So if we look at these molecular models over here, these are what are called space-filling models. They're exactly the same thing as the drawing that we saw before. These are phospholipids. Phosphate head here, here's saturated fatty acid tail, unsaturated fatty acid tail. The other phospholipid, so there's phosphate head, fatty acid tail, fatty acid tail. Okay, so again, the fatty acid tails are sandwiched in between in this hydrophobic environment. The hydrophilic phosphate heads are on the outside, interacting with all of these water molecules on the outside. Okay, so that's about it. For now, that's it for phospholipids. Like I said, when we get into chapter 4 and start talking about cell membranes, we'll talk about this in a little more depth. Anybody have any questions? That's about it for phospholipids for now. Anybody have any questions on that? Okay, everybody kind of understands how the phospholipid bilayer forms. Good. All right, so let's talk about the last kind of fat, the last kind of lipid rather, that we're gonna consider are called steroids. Okay. Steroids are lipids with 4 fused carbon rings. Okay, so we can see them here. So 1, 2, 3, 4. They all share that structure. So we have different kinds of steroids but they all share this fused four-ring structure. These can be components of cell membranes, or they could be hormones. Can anybody tell me before we continue, what exactly is a hormone? What's the function of a hormone in an animal's body? Balance, in a way. So anybody, let me see, I'm neglecting the Zoomers. Anybody in chat on Zoom want to chime in on that? Exactly what is a hormone? So, send signals. Okay, so hormones are chemical messengers. They transmit signals in the body. So let's look at some examples. So we have 3 steroids over here. Now, cholesterol is a component of cell membranes. In recent years, cholesterol has gotten a bit of a bad rap. People say, oh, your cholesterol's high, you've got, eating cholesterol is bad. Cholesterol is actually very important in the right amounts. It's very important. We'd be very sick. Our cell membranes would not be right at all if we didn't have cholesterol. So cholesterol is a very important component of cell membranes. Again, we'll get into that next chapter, chapter 4, when we're looking at cell membranes in more detail. Okay. Down below, we have 2 other hormones. We have testosterone and we have estrogen. So these are the male and female sex hormones. So what do testosterone and estrogen do? Well, during puberty, young males are going to start to produce, their pituitary glands are going to start to produce testosterone. And this is going to trigger expression of secondary male characteristics. Okay, so this is, in adolescence, this is when males, they start growing broader shoulders. They might start growing a little hair on their face. They start expressing secondary male sexual characteristics. Okay, same thing for females. When they go through puberty, they start expressing estrogen, and they'll start expressing secondary female characteristics. So hormones, again, they can be components of cell membranes, as cholesterol. 2 examples of steroids being hormones. We have testosterone and estrogen. Okay, here's an example of cholesterol as a component of cell membranes. So cholesterol kind of inserts itself in between the phospholipids, specifically in between the fatty acid tails. Okay? And so this fused 4-ring structure, this is hydrophobic, so it can get in between the fatty acid tails. This part right here is a hydroxyl group. Okay, hydroxyls are a polar functional group, so they're going to want to interact with the hydrophilic fatty acid, the hydrophilic phosphate heads, rather. Okay, so again, we look at cholesterol. See, there's a hydroxyl group here. Okay, hydroxyl is that's polar, so it's going to want to interact with the phosphate head, and the rest of the molecule here, very hydrophobic. It's going to interact with the fatty acid tails. All right, so it inserts itself like this, with the hydroxyl here, 4 rings interacting with the fatty acid tails. Now, that fused 4-ring structure is planar, so it's very flat and very rigid. So it can help to provide some structural support to the fatty acid tails in the phospholipid bilayer. Okay, so that does it for lipids. Anybody have questions on any of the lipids we talked about? Fats, phospholipids, or steroids? All right, if there's no questions on that, let's talk about proteins. Proteins are the most diverse class of biomolecules. So the monomer or the building block that we can fuse together to make proteins is called an amino acid. There are 20 different amino acids. So because we have 20 different amino acids that we can combine in different combinations, that makes proteins very diverse. So we can, there's all kinds of different things we can do with those 20 amino acids to make different proteins. Proteins are the most diverse class of biomolecules. Proteins are polymers of amino acids. And all of the amino acids have this same basic structure or the same general structure. Okay, so 4 things common to all amino acids is that we have a central carbon atom. This is sometimes referred to as an alpha carbon. So that's I apologize for my penmanship, I'm trying to write with a mouse, but that's my attempt to write the Greek lowercase letter alpha. So alpha carbon. Okay, so the alpha carbon is the central carbon, which is right here. Second thing common to all amino acids is an amino group. Okay, so hopefully, I forgot to mention that on your quiz today, I'm going to start asking about functional groups. Hopefully you guys have been studying your functional group handout. So the amino group is over here in red. Carboxyl group, by now I hope you guys should recognize this as well. So the carboxyl group is over here in green. And R equals a variable side chain. There are 20 different variable side chains, okay? 1 side chain for each of the 20 different amino acids. Okay, so R is going to be one of 20 different variable side chains. So here are the 20 different amino acid side chains. Okay, so you can see these can be very simple or they can be quite complex. The simplest R group or side chain is simply a hydrogen. Okay, so that's glycine has a side chain that's just a hydrogen. That's the simplest of the 20 amino acids. Okay, or we can have rather large and complex ring structures like in tryptophan. So tryptophan has a pretty large and complex side chain. Also notice they have different properties. We have positively charged amino acids, lysine, arginine, and histidine. Negatively charged amino acids, aspartic acid, glutamic acid. We have aromatic groups, that these have hydrophobic side chains. Non-polar, these are also hydrophobic. Polar, these are hydrophilic. Positively charged, negatively charged, these can be hydrophilic as well. Okay, so we have a lot of different properties of these side chains. And again, that contributes to the diversity of proteins because we can make a lot of different things by combining these in different combinations. Okay, amino acids are joined by peptide bonds into polypeptides. The sequence of the amino acids is encoded in DNA. Okay, so the sequence of the proteins we join together with peptide bonds is encoded in DNA. All right, so you guys might recall, when did we talk about this? I think it might have been, no, it was earlier in this PowerPoint presentation. So we have 2 amino acids here. We can join them in through join them together to make a dipeptide here. Does anybody recall, what's this type of reaction called? So it says dehydration here, but we used a different term earlier. So when we take 2 smaller, lower energy molecules, join them together to make a bigger, higher energy molecule, produce a molecule of water in the process. What kind of reaction is that called? Polypeptide. We did this earlier. So remember, the opposite, when we take a bigger, higher energy molecule, break the polymer apart into monomers, get energy out, and consume a water molecule, that's called a hydrolysis reaction. What's it called when we join them together and make a water molecule? Nobody recalls this? Okay. Anybody on Zoom? I haven't been looking at the Zoom chat. Nobody's has anything to say on Zoom. Okay. This is called a condensation reaction. Okay. Remember, we went over this earlier. So we take two smaller, lower energy molecules, join them together to make a bigger, higher energy molecule, produce a molecule of water in the process. That's a condensation reaction. Okay. And I also mentioned that this is an endergonic reaction. What did I mean by endergonic? You guys recall that from last lecture? So if a reaction is endergonic, does that mean we're putting energy in or getting energy out? Putting energy in. So this is a biosynthetic reaction, endergonic reaction, where we're building bigger molecules. Okay, and again, that's called a condensation reaction. Okay, so polypeptide chains or chains of amino acids linked together by peptide bonds can fold into complex three-dimensional structures. Okay? And this is going to be largely determined by the sequence of amino acids. So looking at this figure below, each of the black dots, so these sort of beads on a chain represent amino acids joined together by peptide bonds. Okay. The black dots represent hydrophobic amino acids, and the white dots represent hydrophilic amino acids. Okay. So if you look at the white dots, so remember that hydrophilic wants to be around water. Hydrophobic means not wanting to be around water. So the hydrophilic amino acids, you think they're going to want to be on the surface of the protein interacting with water, or at the core of the protein where they're away from the water? Anybody? So if you just, we'll look at the picture. So we look at the picture, the white or the hydrophilic amino acids are all out here on the surface. Okay, so hydrophilic means, liking water. So they're going to want to be on the outside of the protein where they can interact with water. Now the hydrophobic amino acids are going to want to be away from the water. So they're going to be down in the middle, in the core of the protein, where they don't have to interact with water molecules. So here you can see all of the black, or most of the black, hydrophobic amino acids are kind of tucked away at the core of the protein where they don't have to interact with water. Okay? So there's actually many chemical interactions that are going to determine the three-dimensional shape of a protein, but this is one of them. Okay, so hydrophobic amino acid residues wanting to be in the middle, hydrophilic wanting to be on the outside. This is one of the forces that's going to determine the three-dimensional shape of a protein. Okay, so let's talk quickly. There's actually 3 levels of protein structure. Amino acid bonding patterns form secondary structure in proteins. So first let's talk about exactly what primary structure is before we talk about secondary structure. Okay, and I think I want to go back to the whiteboard for this. Okay, so primary structure. And oftentimes primary will just kind of abbreviate a 1 with a degree symbol like that. That means primary. Okay, so primary structure, when you write primary like this, is simply the sequence of amino acids in a polypeptide. Okay, and again, like the PowerPoint says, this sequence is determined by DNA. Okay, then there's secondary structure. Okay, so secondary structures are areas of the protein. So these are localized structures stabilized by hydrogen bond. So amino acid bonding patterns can form secondary structure in proteins. There's two types of secondary structure, alpha helices and beta pleated sheets. So look over here on the left-hand side. This is an example of an alpha helix. An alpha helix is just like it sounds like, it's just a right-handed spiral. A beta pleated sheet is where we have these structures called beta strands that join together to make a flat surface. So here's an alpha helix. If you can imagine, so alpha helices resemble a spiral or a spring. So if you've ever had like a slinky toy that kind of looks like an alpha helix, if you've ever taken the slinky toy and stretched it out so far that it wouldn't spring back again, you kind of have a piece of metal that kind of looks zigzag like that. Okay, that's kind of what a beta strand is. These are beta strands. So when we stretch out an alpha helix, we get kind of this zigzag. So we can take a lot of those beta strands, join them together side by side, and make a flat surface called a beta pleated sheet. Okay, so we have two types of secondary structure, alpha helices and beta pleated sheets. Let's go back to the whiteboard for a little bit. So secondary structure, we have alpha helices. And again, there's that little squiggle, that's the Greek lowercase letter alpha. And we have beta pleated sheets. Okay, so it's primary structure, secondary structure. We talked about tertiary structure a little bit. So tertiary structure is simply the three-dimensional shape of the protein. So on that slide where we talked about how the hydrophobic amino acids want to be in the middle and the hydrophilics want to be on the surface, the three-dimensional shape that the protein took on after that, that's the tertiary structure of the protein. So the tertiary structure is the three-dimensional shape. Okay, so there's three levels of protein structure. Primary structure, the sequence of amino acids. Secondary structure, we have alpha helices and beta pleated sheets. And then tertiary structure is the three-dimensional shape of the whole protein. Okay, and if we look at this, so let's have a look at this protein here. So this is what's called a ribbon diagram of a protein folded into its tertiary structure. So we can see within the tertiary structure in this ribbon diagram, there are alpha helices and beta pleated sheets. Okay, so highlighted in blue in this square here, we can see an alpha helix, and highlighted in this square here in blue, we can see a beta pleated sheet made from 3 beta strands. Okay? All right, and I don't think we have enough time to get into denaturation, so let's stop there. Again, let me remind you, quiz number 3 will be online today by about noon. Remember, test number 1 coming up February 15th. I'll put an announcement on eCampus so everybody knows. And I think I'll also remember that because we're off on the 11th for a non-instructional day next week, there will be no lab next week. All right, I think that's about it. So you guys have a good weekend. If there's no questions from the people joining by Zoom, I'm going to end the Zoom meeting, and I'll see you guys next week.